As we all know, a cell is the basic structure of all living organisms. Cell groups form into tissues which then turns into organs. But what is the important task for a cell? With Francis Crick and James Watson, a clever duo, they discovered and proposed the central dogma in 1958. Central dogma explains the flow of genetic information from DNA to RNA to make a product known as protein. Central dogma have three processes: replication, transcription, and translation. These three is used by all cells to maintain their genetic information and also to convert the genetic information encoded in DNA into gene products, either RNA or protein, depending on the gene.
What is DNA? DNA, or deoxyribonucleic acid, is the hereditary materials in humans and all other organisms. The information in DNA is stored as a code made up of four chemical bases, which is the A, G, C, T, or A stands for adenine, G stands for guanine, C stands for cytosine, and T for thymine.
The first process of the central dogma is the DNA replication. DNA replication is the process by which the DNA makes a copy of itself during cell division. The first step in DNA replication, an enzyme called helicase breaks the hydrogen bonds between the bases of the two antiparallel strands. The strands are initially split apart in areas that are rich in A and T base pairs. There are only two bonds between adenine and thymine, forming a replication fork. The second step, RNA polymerase, also known as RNA primase, synthesizes short RNA nucleotide sequences that act as primers or starters. These essentially provide a starting point for DNA replication. For the third step, DNA polymerase III can now start synthesizing the new DNA strand using the free DNA nucleotides. However, DNA polymerase can only read the original template or the parent strand in the 3 to 5 direction. This is not a problem on the leading strand because the DNA polymerase can simply continue to read along as the two parent strands continue to unzip. For the fourth step, on the lagging strand, DNA polymerase moves away from the replication fork. As the strands continue to unzip, more DNA is exposed and new RNA primers must be added. As a result, the lagging strand is synthesized in short bursts as DNA polymerase synthesizes DNA in between each of the RNA primers. The fifth step, the newly synthesized lagging strand now consists of both RNA and DNA fragments. The DNA fragments are known as Okazaki fragments, after a Japanese scientist who noticed that heating DNA during replication, which separates the strands, gave many small fragments of DNA. From this, he concluded that one strand must be synthesized in short bursts of DNA. The second to the last step, DNA polymerase I now removes the RNA primers and replaces them with DNA. And finally, for our last step, DNA ligase joins the DNA fragments of the lagging strand together to form one continuous length of DNA.
Let's talk about RNA. The central dogma of molecular biology describes the two-step process, transcription and translation, by which the information in genes flows into proteins, DNA to RNA to protein. Transcription is the synthesis of an RNA copy of a segment of DNA. RNA is synthesized by the enzyme RNA polymerase. RNA is a nucleic acid which is made of nucleotide and is a linear polymer. Each unit or monomer is only attached to at most two other units. They are connected in a series and make it a linear polymer. The specific sequence sequence of each of these monomers is effectively encoding information, and now transfers of information faithfully preserved from DNA to RNA to protein. Each polymer is used as a template for the synthesis of the next polymer. You could go in any step of this sequence and determine what is the corresponding polymers looks like. You can take DNA and figure out what the corresponding RNA looks like. DNA can be copied into RNA in a process called transcription. Transcription is the process by which the information in a strand of DNA is copied into a new molecule of messenger RNA or mRNA. Transcription, from the word script itself, is a one written form to another written form. DNA and RNA are both nucleic acid, and they both use the sort of alphabet. Formation of RNA from codes written on DNA is known as transcription, where DNA double helix and zips and unwinds. Then there are free ribonucleotides which pair up with the complementary bases of one of the DNA strand. The sugar and phosphate of the neighboring ribonucleotides keep joining, and sugar-phosphate backbone of RNA is formed. This pairing up of complementary ribonucleotides along bases of DNA strand is monitorized by RNA polymers, an enzyme. As at the end of this pairing up process, a new single-stranded RNA is formed. The newly formed RNA may undergo processing and will later be used for protein synthesis. Translation.
Let's talk about translation. In molecular biology and genetics, translation is the process in which ribosome in the cytoplasm or endoplasmic reticulum synthesize protein after the process transcription of DNA to RNA in the cell nucleus. The entire process is called gene expression. DNA info is copied into the RNA code, which is still in the language of nitrogenous bases, except adenine on the DNA pairs with uracil in place of thymine on the RNA. This happens in the nucleus. Then, the RNA code is then translated to protein code, which is a different language. This process involves ribosomes and two kinds of RNA, mRNA and tRNA. This happens in the cytoplasm. Directionality of the mRNA. There are two ends in our mRNA, the 5' end and the 3' tail. The 5' end has a cup of methyl group. This end is also called the methyl cup. The 3' tail has a continuous segment of the A nucleotide. This region is called the poly-A tail. And now we have the ribosomes. Ribosomes act as a molecular machine for creating protein. It attaches proximal to the 5' end and then attracts the tRNA. On the other hand, a tRNA molecule has an L structure, held together by hydrogen bonds between bases in different parts of the tRNA sequence. One end of the tRNA binds a specific amino acid, and the other end has an anticodon that will bind to the mRNA codon.
During translation, the mRNA is read according to the genetic code, which relates the DNA sequence to the amino acid sequence in protein. Translation occurs within all cells. The translation machinery resides within a specialized organelle called the ribosome. In eukaryotes, mature mRNA molecules must leave the nucleus and travel to the cytoplasm, where the ribosomes are located. On the other hand, prokaryotic organisms, ribosomes can attach to mRNA while it is still being transcribed. In this situation, translation begins at the 5' end of the mRNA, while the 3' end is still attached to DNA.